session. What I'm going to do is I'm going to give a short talk, an introductory talk about the courses in general, and I'll go through that quite quickly because you might have seen that before, or similar slides at an open day. And then the taster session is essentially going to be a talk with different members of staff and different students to give you an experience of what it's like to take a course in biosciences at the University of Wolverhampton. So I'm just going to share my screen now. Sorry, it just took me back to the main room. So hopefully you can see this slideshow. Well, let me know if you can see the slideshow so I can start, because I can't actually see you now.
Yes, Dr. Connor. Yeah.
That's great. Thanks very much. So welcome to our biology courses. As I said, I'm going to go through these slides quite quickly, but feel free to ask any questions by unmuting your mic or putting it in the chat. I can't actually see that, so if it's urgent, I'm sure someone will pick that up, or we can answer them at the end. So our courses are really 5 main courses. They consist of biochemistry, biological science, genetics, molecular biology, microbiology, and biotechnology. And we also run an HND applied biology. But these 4 main BSc courses, we also run the sandwich courses and these courses with a foundation year as well. There's also the opportunity to do a master's course following your undergraduate course. I think the main theme of our university, the core theme of it, is a number of things in terms of our unique selling points. And I think the main one is that it's underpinned, our courses are underpinned by practical work, laboratory-based work, which is applicable to the world of work. So there is a high practical content in this, and you'll discover this when we talk to the other members of staff and students who I'll introduce as we go along. But all of this high practical content is underpinned by research. So most of our lecturers actually do research, and they publish on this research, and you'll hear more about that as they give a little bit of a talk about their research and their teaching. All of our courses in biological sciences and chemistry are now accredited by the RSC or by the RSB, Royal Society of Biology. And I think that because we offer this high practical content with these transferable skills that are transferable to the world of work, these employability skills, this means that we get really high employability percentages or rates. So a lot of our students go on to graduate level jobs. I think another unique selling point of the university is the amount of support you get. And I think hopefully you'll hear this from some of our students when they talk in a moment, that we do offer a lot of support. It's not a case of you go to a lecture or you have a tutorial or a practical and that's it, you don't get to speak to staff members anymore. I would say that our doors are always open. And you have lots of different levels of support, from module levels, lecture level supports. We have teaching associates, graduate teaching assistants. And we also offer our support through a number of different, I suppose, ways, such as our virtual learning environment, or our VLE, and our student booking system called SAMS, where you can actually go online and members of staff put dates available for meetings, and you can book these meetings on a one-on-one or a group meetings when they're available. But you'll probably find that as you do get to know staff members, that you can just often email staff members and they will speak to you. They will often speak to you in our shared offices, and students often come into the office if they're passing by just to say hello or some help or support with some of their work. So we do offer a really high level of support and guidance. And there is also general academic counselling, student support, personal tutors, course leaders, and they are there to help you with sort of less academic and more pastoral support. And you can see there's lots of other support, and you can go and chat about that on the Chatify at 6 till 7 later on today. So I think that's the areas of our courses that we kind of are proud of and form our kind of unique selling point. When you actually come to study here, the amount of time you'll spend on studying, it does kind of depend really on which course you're doing, but as a general guide, you would spend 12 to 16 hours contact time per week. And this means that this is either face-to-face or live online contact time, or sort of tutorials, lectures, practical classes and seminars. And you would be expected to study 20 hours of your own time by background reading, writing, assessments, that kind of thing. And we assess in lots of different ways, because people like to be assessed in different ways, they learn in different ways. And therefore we use a variety of different assessment types, as shown here, from standard exams to practical reports, presentations, group work and portfolios. There are lots of opportunities at the University of Wolverhampton, from work experience to field trips, industrial visits, lots of careers events. There's opportunities to get involved in the course as well and represent your fellow students as a course rep or in societies. There's science, technology, engineering, mathematical ambassadors. There's opportunities to undertake a sandwich placement, which Dr. Gibson is going to talk about a little bit later. And the opportunities then for your career are really wide-ranging, from ones you might think about, such as pharmaceutical companies, medical research, biotechnology, biomedicine, to ones that are a bit more unusual, like agrochemistry, waste management, and then there are lots of opportunities for water authorities, testing water, and in the food industry, it's a huge industry there. Even things such as marketing, sales, management, and public service. Things like medical communication or writing is a career that's applicable to these courses. And then there is also postgraduate research and teaching, which we'll talk about a bit later with some of our students. And I think this is reflected in our high percentage of employability, of graduate employability, because these are hardcore courses. These are science courses. These are going to open doors to the world of work. So why choose the University of Wolverhampton? Well, I think that, as I said, that I suppose unique selling points, as well as the fact that our courses are all accredited with Royal Society of Biology accreditation. I think that one of the main things is that we have a high level of research informed teaching. We have lecturers who actually publish research and then they teach you. So they are doing the research and then teaching you as well. And not only are they actually carrying out research, but they are publishing in peer-reviewed papers. We also have state-of-the-art facilities. I'm going to show you a short video in a second about that. And in those state-of-the-art facilities, you do really up-to-date practicals. These aren't labs that have been going on for 20 years. They're modern, updated practical classes that are applicable to the type of thing that employers want you to do. So you're learning these skills and you're learning these employability skills alongside the technical skills as well. And I think with all that, with this amount of support, guidance and feedback you will receive at the University of Wolverhampton, I think this will really stand you in good stead for your undergraduate career and beyond. And why do why do we do all this? Well, I think at the end of it, you want to graduate with your degree and go on to your next stage of your career. And I know this might seem a bit premature to put this up now because you're just starting out on your university career, but this is what it's all for. We want you to graduate, and these are some of the pictures, the sort of formal university pictures of graduation. But graduation to me is more like this picture. These are some of our fellow students. There's a few pictures of the last couple of graduations with myself, you'll see there, and Dr. Mark Morris in some of these photos. And this is actually the best day of the year for me, really. It's the culmination of 3 years hard work and moving on to the next stage. It's the time when we often get to meet your families for the first time, friends and families for the first time, to celebrate your graduation. And all these students have gone on to various different areas of work. So some work in industry in Birmingham and Wolverhampton and further afield. Some go into teaching, some go into research. There's a number here that have gone on to do PhDs. Some go into medical science writing. So there's a wide variety of jobs that they go into. But I think one of the key things is, I know I keep stressing this, is the idea that the university or the University of Wolverhampton has lecturers who teach you, who do research, because that's what a university is. If you look it up in the dictionary, a university is a high-level educational institution that has the power to confer degrees. So that means that we can give out our own degrees. But what's crucial is where research is carried out. And without this research and research-informed teaching, then it's not really a university. And I just want to show a quick video of where, if you choose the University of Wolverhampton, where you will actually carry out a lot of this practical classes and research. It's just a 5-minute video, so I'll just click on this and hopefully it should play. If not, I'll share the screen in a different way. To the start. Can you see and hear this?
No, Matt, nothing there.
No, nothing at the moment.
Okay. 1 second. Okay, hopefully that should work.
Looks better, yeah.
Is that okay?
Hello, I'm Dr. Martin Cashel, and this is the University of Wolverhampton. And we are standing in the fantastic Rosalind Franklin Science Building. You are about to witness the £25 million investment in science teaching the university has made. And we're here today to actually meet a very special scientist. But that scientist is all the way up on the very 5th floor of this building. So what we do, what I thought we'd do at least, is just have a little tour around the building on our way up there before we meet the fantastic Professor Isabella Wiedecka. And here we are on the very first floor of the Rosalind Franklin building. The Rosalind Franklin building is actually divided into subject areas depending on what sort of floor you're on. So here we are on floor 1. Now, on the first floor, what we have is chemistry. So behind me, you can see that this laboratory is set up for a practical session, ready to run face to face, even in spite of our COVID pandemic, about a subject in chemistry. Logical practicals actually happen. Now here we are on the second floor of the Rosalind Franklin building. Now if you remember what it looked like before, on the first floor, which was primarily involved in things like chemistry, actually now on the second floor, we're involved more in biological sciences. And here on the second floor, we do things like biomedical science, hematology, anything really, microbiology, anything to do with the things that infect a person or the flu. We teach people how to analyse those sorts of samples to be able to actually diagnose disease in real patients. And you can actually see behind me that the laboratory is set up in lots of different ways. You can see we've got microscopes behind us and so on. And also you might see that some of the desks have got tablet devices on a sort of arm. And actually, as students, we can actually get 90 students in here at a time, when we're not in a lockdown, of course. So students get a tablet between two, and that allows them to record their data in real time without paper straight into their sort of student learning environment, which is absolutely fantastic. So we have people from all over Europe come and actually see the sorts of things that we're doing here in the Rosalind Franklin building, because it is considered best practice for science education. But this isn't all, because we're just on the second floor and we've got 3 floors left to go. So here we are on the 3rd floor of the Rosalind Franklin building now. Now, the first floor, as I said, was to do with things like chemistry. The second floor was to do with the biological sciences that concerned the fluids of the body and the infectious things of the human body itself. But this floor is about biochemistry and molecular biology, about DNA and about analysis of fluids that allow us to diagnose disease. And I'm talking quietly because over here we can't show you that at the moment. Right now there's actually a class happening face to face, even though we are under COVID regulations. We can still have our face-to-face practicals in a COVID-safe manner in these laboratories. And these students are experiencing how to actually use scientific equipment that is going to be their tools of life as scientists as they move forwards through their career. What we're going to do, we're going to move up to the other floors, and they are very special places indeed, because they concern the research that the university does through the Faculty of Science and Engineering. Okay, so let's go and have a look at the 4th floor. See you later, everyone. Bye. Now here we are on the 4th floor of the Rosalind Franklin building, and this is the absolute hive of the research effort in the Faculty of Science and Engineering. Here there are many different subject areas represented, and there are postgraduate students carrying out their PhD sort of research programs, and also academic researchers here working on loads of different kinds of projects. One of the things we do, particularly in this facility here, is look at new and improved ways to treat cancer. And that's everything from bowel cancer to brain cancer. And every day, new discoveries are being made just in this room. I mean, how fantastic is that? But the scientist that we're going to see doesn't live here. She's one more floor upwards. So let's go and see.
Okay, I hope you enjoyed that. There was a quick whistle-stop tour of our Rosalind Franklin building, all our labs and everything. I'm going to come back to that in a minute, where you will meet the excellent professor that Dr. Kashir was talking about. But first of all, I just wanted to introduce one of our current students, Louise Davis. Hi, Louise.
Hiya, can you hear me?
Yes, we can hear you.
Oh,
that's
good.
great. So I've just asked, Louise has kindly agreed to come on here just to talk about her experience, really, what it's like to be a student. I haven't really briefed her either, so I've told her to tell the truth. Would you like to just give us a bit of a background, just tell them that you're studying biochemistry and what year you're in? That'd be great. Thanks, Louise.
Oh yeah, hi, I'm Louise, everyone. I've just finished up my biochemistry degree now. Did my last exam last week, so I'm graduating this year. Yeah, so that's me, biochemistry.
And you've just finished your undergraduate project, which was obviously a bit different this year because of the COVID restrictions. So how did you find your honours project?
Honestly, it couldn't have worked out better, because even though I was expecting like a lab-based project, I still found it like very fulfilling to do it remotely. But also I did my dissertation with Dr. Mark Morris, which is here. Yeah.
Good.
Honestly,
I've got to hear that. Yeah.
I really did it. I wasn't, honestly, no offense, I didn't expect it to be as amazing to do it remotely. I was skeptical because obviously I like labs. Like that's one of the reasons I loved doing my degree, is because in first and second year I got so much lab time, and that's what I was excited for. But obviously the pandemic, so I didn't have that in my last year, but honestly, right, it was still very fulfilling.
That's great, and it doesn't hurt having the amazing Mark Morris, who you're going to hear from in a minute. Yeah. So would you say that was your best experience, or your favourite? What would you say your favourite lecture was, or practical? Would you say it was that, or was there some others that you could highlight?
Definitely. I mean, going back, we did like in the second year, it was for 003, where we did it was basically like you did like a mini project, and literally the lecturers treated you as if like to treat your own experiment and plan it like you were doing a final year project. So I thought that was amazing in second year, because I was like, oh, I get to like, it was like almost like I was learning a skill that I'd use in my projects, and obviously I would have used that skill if we wouldn't have been in a pandemic. So I thought that was amazing how we had that experience all the way in second year to help you through your third year.
Good. And so have you had any other good experiences, and what if you could give some advice to prospective students, or anything like that, what do you think you would say to people wanting to, or thinking about studying at the university?
Well, I do think that help is there when you ask for it. A university in general, I'm a believer that you get out what you put in. Like if you want to work hard and you genuinely want to ask the questions, everybody at this university will answer you and will help. So basically, as long as you want to learn and put the effort in, you'll always get the help here. From the way Dr. Connor was saying earlier, there's so many ways to contact them, all the lecturers. So that's my advice. You genuinely, you get out what you put in. So as long as you want to learn, everybody will help you do it. Yeah. Well,
that's great. Thank you, Louise. That's really good. I know that Hazel Gibson, Dr. Gibson, has just put in if anybody's got any questions, but would anybody like to ask Louise anything, or you can either unmute your mic or you could just put it in the chat. If not, I think, Louise, are you going to hang around for a little while, or do you have to go? No, that's great. So thank you, Louise. If anybody has got any other questions, just pop them in the chat, or you can ask as we go along. And also for me as well, when I just talked about the overview of the courses and everything, that was a really quick whistle stop tour. So if you've got any questions to that, please feel free to ask as well, and we'll do our best to answer them. I want to introduce one of our lecturers now, Dr. Mark Morris, and I'm going to introduce everybody here. We've got staff members and some students as well, undergraduate and postgraduate. I'm going to introduce them as they go along, and they're going to talk a little bit about what they do within the university, their teaching, research, and their studies. So we'll start off with Mark Morris, Dr. Mark Morris. Hi, Mark. Mark is a reader in molecular oncology, DNA guru. So Mark's just going to tell us a little bit about, you can talk about a little bit about research and your teaching.
Yeah, thank you, Matt. Can everybody hear me? Is that all okay?
Yeah.
Good, you can hear me, great. Yeah, so I've got a couple of slides. Yes, I'm going to talk a little bit about what I'm interested in and then how I kind of bring this into my teaching as well. Let me see if I can get these slides up. Tell me if this is working. So I can't see you guys now. That's not ideal, is it? Is that the way it works, Matt?
It is, yeah. We can't see. I can't see your slides yet. Just give it a second.
Because I haven't started to share my screen.
That'll be why.
How's that?
Yes.
Yeah.
Yes, we can see that. That's great.
Fabulous. And I can see you. That's absolutely perfect. Okay, cool. So before I start, I want to say, Louise, thank you for coming along. And I need to reiterate that Louise did a really nice project. She's made actual discoveries from what appears to be her bedroom during lockdown. So no, proper discoveries that have really contributed to my ongoing research. So she's been a real asset and that's just in her third year project. So really, no, a real cracking project and really genuinely useful and interesting stuff that you discovered. So very, very cool. Okay, I'll get back to that a little bit. So a bit about myself. Let me make this move. There we go. Okay, I'm going to turn on my laser pointer as well. There you go. So really busy screen. Don't worry about too much about anything on here. But I'm generally, I consider myself a cell biologist. Although I'm really a cancer oncologist, a molecular oncologist. I'm interested in the molecular processes of cancer. This is how normal cells go wrong, start to divide too much and grow. So not so long ago, I was an undergraduate, just like you guys are about to become now. And I did a degree in biological sciences. In fact, it was biological sciences with molecular genetics. So it was a bit like a molecular genetics degree. And following my graduation, I went home and got myself a job as a painter and decorator while I continued to apply for jobs after this. You know, I didn't walk myself into a PhD or anything, but then I eventually got a job as a research technician in a research laboratory that was interested in cancer. And I've been very interested in cancer as an undergraduate anyway, the molecular biology of it. And whilst being a technician, I wrote a PhD. You know, I was doing enough of my own independent work to create my own PhD. So I got my PhD while I was working at the University of Cardiff. And then I moved to the University of Birmingham, where I worked under two very famous professors who were very, very much into the molecular biology of kidney cancer, where I stayed there for 10 years. We discovered quite a lot of stuff, did a lot of genomic analysis of cancers and cells to try and work out how they were breaking. And 11 years ago now, I moved to the University of Wolverhampton, and I've continued that work. So I've continued my interest in cell biology, genetics, and the evolution of cancer. And this is kind of my big question. This question I have about how cells go wrong really feeds into all of my teaching, because I think this is the really neat thing about this, is that when you say, what do you need to learn to be a biologist or a biochemist or a molecular biologist? You basically need to know how cells work. If you know how a cell works, there are so many processes in this one system that really informs you about most of biology. So I think it's a really neat way of sorting things out. Then my big question is this, is how do healthy cells become cancerous? And all it really is, it's an incredibly complicated idea, but all it really is that normal cells pick up random mutations as they divide, and these random mutations give them additional ability to survive and grow. And this gets increasingly complex and bad for the person who's got these cancers growing in them. Why this is useful to us as a wider group of biologists is this simple idea that if you want to know how anything works in its normal, correct way, you have to know how it works when it's broken. If I was to give you, if I was to say, how does a watch work? The best way for you to know how a watch works would be to take off the back and start taking things out of it and see how it doesn't work, and then putting the things back in. And that's kind of what I'm doing. I'm looking at cancer to see how normal cells work by seeing how they don't work when they're broken. Okay, so what I'm doing is I'm doing genome sequencing, proteomic analysis, trying to see how proteins go up and down, how genes are mutated, the expression level of all these different things, all at the same time, to try and get a snapshot at different points in the evolution of cancer to see how they're going on. Looking at different cancers as well, so it's all sorts of things going on all at the same time. And this informs what I teach you in the classroom. So back to this basic idea, this simple idea is, what do normal cells look like? How do normal cells divide correctly, differentiate into all these different tissues to make whole areas of tissues, organs, and organisms eventually? And the way we can work out all of this is by looking at how they work, how they don't work when we break them through mutation. And this informs the teaching that I provide to you. So we need to start really at the basics. I start to think about what the structure of DNA is. So I'm going to be giving you some of this stuff right at the beginning in year 4, structure of DNA, but not just the structure of DNA, the kind of more complex, how DNA is packaged into the nucleus and how this packaging enables access from enzymes to the DNA to allow transcription of RNA, to allow translation into proteins. And then we're going to think about how DNA encodes these complex 3-dimensional proteins and how these 3-dimensional proteins are the functioning units of your genes, and how when you get mutations, these proteins don't work, and how it's all linked back to cancer. And then really, this is what I'm really interested in, and this comes in an awful lot, is, you know, we get through the basics of DNA, transcription, translation, and really it's how do cells talk to each other and talk amongst themselves. It's this idea of complex proteins such as these cell surface receptors sitting on the cell surface, binding to external factors, growth factors, which then change the shapes of proteins, which then allow them to bind to other proteins, which then change the shapes of these proteins, which then bind to other things and modify those. And eventually it works its way all the way down through this complex cell signaling pathway. This is the center of how cells work, to eventually tell the DNA to divide to allow the cell to then replicate itself. So cells replicate by dividing all of their DNA and dividing all the protein and all the lipids, all the organelles in the cells. And this is a complex, orchestrated program, which is incredibly precise. And when it breaks, then we start to go back to what I'm interested in my research, which is the formation of cancer. So you get all of this. I'm going to talk about my research in relation to this, but I'm not going to just stick to what I'm doing. I'm going to tell you about the famous scientists, the huge, big discoveries, the Nobel Prize winners. We're going to bring all in to the basic molecular biology that you need to know. Obviously, then you get to your DNA replication. We'll talk about DNA replication and errors in DNA replication, which introduce mutations, which change the structures of the proteins, which bring us back to this idea of how normal cells start to become cancerous, which is the thing that I'm particularly interested in. We will then think about what we're going to do in the laboratory. There's lots of lab work in the next 3 years. And the starting point of this is actually biotechnology and bioinformatics. We're going to use the resources available to us because of enormous progress in the last 15 to 10 years in DNA sequencing technologies and a lot of other molecular biology. There are repositories of so much information online now, which we can go and just grab, and as long as we have the background understanding, we can go and use these to do our own investigation. So I'm going to teach you about how to go and use human genome browsers. This is the genome project, which was finished in 2003, the most recent update. I made this slide today. The last time it was updated was December 2013. It's a finished project. Using this sequence, we can go and amplify any piece of DNA we want. Then we can manipulate that DNA into any kind of other molecule we want. We can express any protein we want in any system, animal cells, human cells, bacterial cells, whatever you want. So we can do all that, and we do all that in the laboratories. So we're going to do some actual biotechnology. We're going to do some gene cloning. We're going to amplify a gene and clone it into plasmids, amplify it in bacteria, purify it back out. We're going to do polymerase chain reaction. We're going to look at protein expression, and something that Matthew and Louise may not have had experience of yet, but something that is coming on board as you guys come through. We're going to start thinking about gene editing using CRISPR systems. This is something that my PhD students are currently doing, where I'm currently knocking out genes that I think are important in cancer, using CRISPR-Cas9 systems. So there's a huge bunch of stuff that's related to my research that comes into the teaching that you're going to receive. And I want to end with this really. In addition to your textbooks and just these ideas, the important thing I think we spend a lot of time introducing you to as well is, there's no point just telling you this is the fact. We need to be able to tell you, how do we know this is the fact? And where do we get this information from? You know, this whole idea of standing on the shoulders of giants, these tiny incremental additions of information which we get, starts way back in time. And what are these key experiments? You know, how can I manipulate DNA without really thinking about it anymore? It's because of the work of Watson and Crick and Franklin and Erwin Chargaff. How do I understand how proteins work? It's because of the work of Linus Pauling and many others. How can I knock out a gene without really thinking about it anymore? It's because of the work of Jennifer Duand and Emmanuel Charpentier from their CRISPR manipulations. These guys won their Nobel Prize last year. Yeah. How can I do PCR? This guy, Carrie Mullis, invented PCR. We go through these basic experiments so you understand how these experiments work. We repeat some of these experiments. You're going to do them in the laboratory. You're going to repeat experiments that I've done. You're going to do your own independent experiments. And it builds to a point where you have a deep understanding of molecular biology, also a good understanding of the techniques involved, and an understanding of where we came from to get to the point we are today, so we can then move forward as we continue on. So I'm happy to take questions, but that's kind of I think that's it. Oh, one last slide. I just want to reiterate that these ideas are then reinforced in the laboratory. You're going to come into the lab, you're going to do gene cloning, you're going to do PCR, you're going to do protein analysis and all these other things. It's not I'm not going to give you a textbook and leave you for 3 years. You're going to be in the lab every week doing something exciting. Okay. I think that's enough. Yeah, I think I probably overspoke.
No, that's really good. That's really interesting. I think I would say that Mark's definitely showed there how you know, he's a researcher who actually teaches and brings that research into the practical sides, into the labs. And one thing, like all the lecturers here today, Mark explains incredibly complex molecular biology in a really easy to understand way, I think. And I really enjoy, lots of the other students enjoy his science history lessons. I'm the only guys that
some of them do.
links the Kardashians to the discovery of PCR in a really nice way. So you'll have to come here and listen to those lectures because they are fantastic. Has anybody got any questions for Mark? And they can be general about teaching or time, or they can be something really wide-ranging. Put him on the spot if you can.
Anything at all. I'll field anything at any time. I think that's I hope, Matt, more than anything today, I hope that you and I and the other staff who are here today kind of can show that we are super approachable. We're not weird nerdy scientists, are we? Yeah, we are.
Well, we are. We're quite, yeah, we're definitely geeky. We publish in all these. Yeah,
but we're reasonably approachable.
Well, I think so. And the fact is, with science today, we should be showing you in the labs. You should have come into the university, but we can't. We would have shown you PCR and lots of different techniques and been there as well. But hopefully this method shows you our kind of teaching style, the way that we speak, and we try and make things simple, and we are approachable as well. So yeah.
It is complicated. There's no doubt, though. To reiterate, Matt, this is hardcore. You've picked a hardcore subject. Well done. This is a good thing you've done. You've already taken the best choice you will ever make by doing a proper subject.
Anybody got any questions? Mohammed?
Yep, I do have a question. Can you hear me, by the way?
Yes.
Yes.
Perfect.
Oh, okay, that's good. I just wanted some more information on the actual course in itself, because what it is, I've applied to do the genetics. That's something that I want to do moving forward. So I just kind of like want to know, what can I expect in the first year, second year, and third year overall as a breakdown kind of thing? How many modules will I be taking in the first year, so on and so forth, things like that.
Matt, shall I leave you to take that as course leader?
Yes, that's fine. So essentially, you will take, in the first year, you will take 6 modules, and that's 120 credits. So each module is 20 credits. So you'll do 3 modules in the first semester, and then you will have your exams at Christmas, or you'll hand in your assessments, which might be write-ups or presentations, lots of different types of assessment. And then you'll get your marks soon after that. And then the next semester will be your second 3 modules, and that will be another 120 credits, and then you will hand in your coursework and have your exams in the end of May, and then hopefully that should be finished. You know, there are retake opportunities later on in the summer if people are ill or whatever happens, and they have to retake. But essentially, that takes place every year. So you have these 120 credits, 6 modules, 3 in the first semester, 3 in the second semester, right up until the third year. However, your project is like a double module, so it's a 40 credit module. And in terms of the genetics course, if that's one you're interested in, like most of the courses, Biosciences, there is a huge an overlap in terms of the fundamental basics. Although there's nothing basic about it, it's complex, but there is a large overlap. Things like macromolecules of life and the basic mechanisms of biology. So in the first year, you will have some specialized subjects with genetics and maybe biochemistry. There will be some others that are shared between biology, biological sciences, microbiology, biomedical science. There's some overlap there, and pharmaceutical sciences, so that you will get this kind of fundamental knowledge of biology. And as you go through your university career, these will specialize more and more into a specialized genetics and molecular biology course, so that there are standalone courses for geneticists. But as I said at the start, you do have some more sort of general fundamental courses as well. And as I said, there's lots of practical based courses as well. I think Hazel might talk about one of her modules, I don't know, which is a great first year module. And then obviously as you get on, you specialize as well. But even if you're in a practical class and there might be people taking biochemistry in that class, then there are opportunities to specialize within those classes. You might be dealing with different molecules based on your degree. And then in the third year, it's much more specialized, and obviously your project is extremely specialized then, because you choose that with the supervisor, and that's when you're doing actual real research. So the list of modules is on the website, or you can email me, or I can put that up for you and get that actual list of modules. I don't think I've missed anything. Does that answer your question?
Yeah, that does. You know, I do have like 1-2 other questions as well, but I can save that for later if you know.
There should be time. There should be some time right at the end, hopefully, if we don't. If we go on too much, and yeah, and I would encourage you to ask them then, and we can go through them then. But thanks for that. Yeah,
no problem. Thank you.
Thanks, Mohammed. Any other questions? Okay. So I think I'd like to introduce you now to another one of members of staff, Dr. Hazel Gibson, who's going to talk a little bit about her research and teaching and general responsibilities. Thanks, Hazel.
Hello, thank you, everybody. I will warn you, I'm struggling a little bit with my broadband. So if it starts breaking up, somebody please tell me so that I'm not, disconnected or frozen or you can't hear me. Then somebody let me know.
Okay.
Hope, fingers crossed it'll behave itself. So I'll just share my screen if you bear with me. Okay, can you all see that? Is that okay?
It's starting. Yeah, it's good now.
Okay, thank you. All right, so I'm just to introduce myself. I'm here. I will just give a little bit of background in the same way that Mark did. Is that I started out on a biological science degree, and I did that because it gave me the area of biology I was interested in. And as part of my biology degree, I was fortunate enough to undertake a number of placement opportunities, which is something that you have available to you in your course, and I'll say a little bit more about shortly. But as part of my degree course, I undertook a microbiology placement, and following that microbiology placement, after I'd finished my degree, that particular placement provider, that research institute, offered me a PhD in microbiology. So that's when I moved and became more specialised as a microbiologist. And that research institute in this area first started. So I'm a microbiologist through and through now. And I teach microbiology, but general microbiology at all levels of the degree course. And I also teach microbiology for postgraduate students. And my particular area of interest is on food microbiology. Now, we had a question about what's involved in each part of the course. And I've listed here just some examples of areas where I teach. In the first year, we have a module that's Introduction to Biosciences, and students on any of our degree courses will study that module, and it's an introduction to both theoretical aspects, but also key to that particular module is becoming familiar with being in the laboratory and familiar with some of the key techniques, microscopy, some of the spectrophotometry that we use. So it's an introductory module to studying at the university. Now that is an example of a module that has some in the lab. And what you find with our courses and with our modules, some modules are theory only, some modules are practical only, and some modules have a mixture of both theory sessions and practicals. And as we've all sort of mentioned already, is that we think that the practical that we have in course is absolutely fundamental to your understanding of the topic forward on graduation. We have other modules, and this is an example. Applied Environmental Microbiology is a second year module, and this is a module that includes theory sessions, but also includes some practical sessions. We have inputs into our modules from external speakers from outside of the university to again give that employer input and insight into the skills and attributes that graduates need to exhibit on finishing their courses. And it also means that our courses are up to date and of high relevance, because we're getting those employers and visiting lecturers coming in as well to enhance our delivery. We've got modules, that is when, as Matt said, that's when it becomes much more specialised. The examples I've included here, because I'm a microbiologist, tend to be more microbiological in nature. But if you're a genetics and molecular student, microbiology and biotechnology student, biological science student, there are different options in terms of what you would be studying in the final year. For microbiology students in particular, we've got a module that's looking at how you investigate foodborne outbreaks, how those outbreaks are managed, looking at the sorts of techniques that would be used to actually confirm an outbreak situation and prove that the strain isolated from a sick individual is the same as the strain that was isolated from that particular food source or that particular water source. So throughout all of our courses, we've got a number of different modules. And as I've said, there is practical modules and practical elements built in throughout your course, ultimately with the project in the final year. Now that's a little bit about my teaching interests have built from my PhD studies and the research links I've had since then. My PhD was looking at attachment and biofilm formation, and this is still an area of research that I'm involved actively in at the moment. We've got a couple of PhD students, and this is a collaborative work with both Aston University, but also with my colleagues in pharmacy. And we've got PhD students who are looking at how to inactivate biofilms, because when organisms attach to surfaces, as we can see in this electron micrograph at the top here, we've got Staphylococcus aureus that's attached to a stainless steel surface. But when the organisms attach to a surface, when they proliferate on those surfaces, as we can see in these confocal microscope images, we can see numerous layers of organisms and prolific growth on the surface. Well, those organisms become very difficult to inactivate. They become less sensitive to antimicrobials. And we've got ongoing research that's looking at how we can enhance the inactivation of those organisms, but also by adding treatments such as different amino acids that enhance the solubility and enhance the action of different antibiotics in the treatment of these biofilms. And these are of relevance, for example, to patients that are suffering from cystic fibrosis, where biofilms are a particular problem in those conditions. I'm also interested in the use of antimicrobials for food preservation, but also novel applications to do with wound dressings and polymers, which I know my colleague Professor Isa Radeka will talk more about, but using things like natural antimicrobials. We've got a PhD student at the moment who's looking at, and this image at the bottom here shows you part of the process of producing yogurt. You can see some clotted milk here, and she's looking at using different herbs and spices, things like cinnamon and cardamom, that's added to this yogurt in this fermented yogurt product. Another area, and we've got a PhD student who's just started recently looking at this, again linking in with my whole interest in inactivation of microorganisms. We've got a PhD student who's currently looking at the pneumococcal vaccine and any relationship and link with the antibiotic resistant strains that we see of strep pneumoniae. So these are all active research projects that are ongoing at the moment, and we're actively publishing in these areas too. Just to move on to another area that I'm involved with is I'm the sandwich placements tutor for this particular department. As I mentioned, I've done sandwich placements myself and benefited both in terms of myself, but I've looked after a number of students that have been on placement in recent years, and I can't emphasize enough, and I know Matthew will say the same when he has a chance to speak in a few minutes, but a sandwich placement year really does enhance your employability and enhance your performance in your degree. If you haven't thought about a placement year, it's open to you, and it's an optional year on placement after completion of the second year of study. It really enhances your CV because you've really reinforced the practical skills and knowledge that you've gained in your degree in a workplace setting. So it's really, really enhanced your knowledge and skills. And we do find that students students that have been out on placement really excel on return to university and often come away with the best degree classifications and come back to university. Now, the workplace environment enhances your knowledge, your skills, but it also is a fantastic networking opportunity. And many, in recent years, we've got students that have completed their placement year, and then they've moved into employment in their placement provider on finishing their degree. And we've got students now, this current academic year, who are working in the setting. Their degree has taken them straight into employment with their placements provider. So it's really, really to be recommended. And as I said, I know Matthew will have something to say about that too. So think about a placement year if you haven't already. In terms of sorts of places you can go on placement, these are just some examples of the companies, institutes that people have worked in recent years. So Innocore is a pharmaceutical research company in the Netherlands. Eurofins is a food and water testing company with a global market with laboratories all over Europe and all over the world. And we've currently got students on placement at Eurofins. We've had students on placement within the West Midlands Police. We had genetics and molecular biology student who had a fantastic experience down at Kew Gardens in London, which was really excellent. We've had students both in a number of hospital settings. We had a Field Studies Council. Now you might think that's more of an environment, who went on placement to that particular environment up in Scotland and was looking at tracking certain species in her placement. The binding site is based in Birmingham, and the binding site have taken a number of our students in recent years on placement, and that's an example of where we've had students actually been taken on as employees in recent years. And this is just a handful of placements that we've had in recent years, but it depends very much on the interests of the student, where they may see their careers going. And we're very lucky here at the university that we have a placements team that are there to help students along the whole process in terms of applying to, going through the interview process and supporting you right the way through until you finally come back to university for your final year of study. So I can't emphasize strongly enough that a placement is something for you to think about. Now, I think I probably rattled through very quickly a little summary of me studying at the university, a bit about my research interests, and a little bit about placements. But if anybody's got any questions at all, then please do ask me. I'm hoping my broadband
Thank you, Hazel.
stuck with me sufficiently for you to be able to hear what I was saying.
Yes, I think it did. Yeah, it was okay. Thank you. Yeah. Has anybody, thanks, Hazel. That was really interesting. Has anybody got any questions for anybody? Just while we're just on that, could I it's just reminded me, asking, it's a question I wanted to ask Louise, and this is a loaded question because I know something about Louise. So would you mind, Louise Davis as well? It just reminded me of that last slide that Hazel put up. What would you like to just tell us what you're hoping to go on to in the next stage of your career, Louise?
Yeah, I did unmute myself. Yes, I did.
Yeah.
I have literally this week started a job with Eurofins. I am a microbiologist in a path lab.
Congratulations.
Thank you. I've just started this week, but obviously going forward, I want to obviously use this to get a couple of years lab experience behind me, and then hopefully self-fund myself in the future to do more research in the future, because it's something I like I said earlier, I enjoyed my project more than I can say, and I want to do that in the future. But pandemic, we all need money, so I can use this experience and go in future.
Well, that's fantastic news. Congratulations. You really deserve it. I mean, you don't have to necessarily use that as a stepping stone. Eurofins is such a huge international company. You know, you could do really well in that company. And they obviously fund their own research and things as well. So something I also would just say on the back of that is that I'm still in contact, and most of us are in contact with a lot of our alumni students, and we help them out, you know, years and years ago, and they're now in jobs, and we speak to them quite often. So that level of support doesn't stop as soon as you leave the university. We're still in touch. We still help them with applications and with their career. Thanks for that, Louise. Well done. That's really good. Really good news. Yeah, well done. Something that that Hazel mentioned as well. She was talking about lecturers visiting lecturers as well. So in terms of your modules, you will have generally a module leader, which will lead on that particular module, one of those 20 credit modules. But within that module, you will have specialist lecturers teaching you a particular area. So you might have a molecular biologist teaching you molecular biology about DNA, or you might have a microbiologist teaching you about something else. We try and keep them reasonably small teaching, so you get to know the staff. But also we have a variety of staff to teach specialist subjects in their area, these research specialities, if you like. And I think on that also, you get also a level of support there with the individual lectures, the module leaders, the course leaders as well. But also we have a number of graduates who now are employed by us to teach, essentially. So graduate teaching assistants, graduate members of staff. And one of those is the next person I'd like to introduce, which is Laurel Chaperonier. Hi, Laurel. Now, Laurel is currently working for us, and she's going to tell you, she's got a dual role really. So if I'd like to ask Laurel to say hi and tell us a little bit about what she's doing and what it's like to work and study at the university.
I am a bit 50-50. Hi everyone, it's lovely to meet you all. So my name's Laurel. I am a student transitions teacher at the university, but I do that part-time, so I teach 3 days a week, and then 2 days a week I am doing a PhD under Dr. Connor. So I'm very lucky to be doing research into aquaporins with Dr. Connor. But my teaching focuses primarily on level 3 and 4. So if you were to do a foundation year or in your first year of study, you'll see a lot of me. I do a lot of the small group tutorials, the one-to-one support if you need extra support on any of the topics, anything like that. That's the kind of thing that I'd be focusing on. But I've been at the university now, this is my 8th year at the University of Wolverhampton, so I don't think I'm going to be leaving anytime soon. I never actually planned to come to the University of Wolverhampton, so I never came to applicant days or anything. I had chosen a different university, and unfortunately my sister became ill. And there was the University of Wolverhampton and a different university that did the degree that I wanted to do, which at the time was British Sign Language. So instead of going far away, I came to the University of Wolverhampton. And even though I decided not to carry on with doing British Sign Language, I loved the university, so I stayed and did a medical science undergrad degree instead. And I completely agree with everything that Dr. Connor said about getting to know your lecturers. Because one of the main examples, and I think that really shows that level of commitment and stuff that the lecturers have to their students, is when I applied for a position as a graduate teaching assistant, which I was before I had this role. I was sat outside, very nervous, waiting for the interview. And one of my lecturers walked past me and I said hello. And we'd had conversations, but I didn't realise how well they'd really got to know me until that point, because they just turned to me and they were like, "Oh, Laurel, like, are you going for the interview?" And I was like, "Yeah." She was like, "You'd be amazing. Like, I completely want you to get that job." And after I came out of the interview, she actually went in and spoke to the interviewers and said that I'd be great for the job. So the lecturers really do have that really deep level of caring for all their students, and I'd never forget what they've done for me. And I went on to do a teaching qualification at the university, and now I'm employed permanently. So hopefully in a few years I'll get my PhD with Dr. Connor and I'll continue on that journey. But yeah, I have good links with the education department. So anyone who's doing biology with QTS, would be quite good to have a chat with you as well, or anyone who's doing foundation year. I have lots of information about the foundation year if you have any questions about that. I'm in my first year as a PhD student, so that's my other half. So I'm in the process of getting all that set up, and I've started going back into the labs, and it's all very exciting at the moment. But any questions about being a student or foundation year or teaching or anything that I can possibly help with, then I'd be very happy to.
Thanks, Laurel. That's really good. Yeah, that's really nice to hear. Laurel's in a unique position, being a graduate, and also is studying for a PhD here. So there's lots of knowledge of lots of different areas, and also as a sort of graduate, I think that sometimes students see her as even more approachable, although I'd hope that we are approachable as well, as we keep stressing. But yeah, it's real credit to the university, and will help you a lot in your undergraduate studies. So if anyone's got any questions for Laurel, that's fine. Just type them in a chat, or you can ask them at the end. That's no problem. We should have a bit of time at the end. And that kind of brings me on to the idea of getting to know your lecturers and making the most of university life, because next I want to introduce someone who I remember coming up in the first year, was at the end of a lot of lectures, asking really difficult questions, and I really enjoyed them, and that's Mattia. And he's actually a postgraduate student here, did an undergraduate study. And would you like to introduce this, or say hi, and just tell us a little bit about your experiences?
Absolutely. First, can you hear me all right?
Yes.
Yeah, that's good. But yeah, you know, when they all say, you know, we're very approachable, and you can come talk to us. Sometimes that's not too good of a thing. I remember Mark Morris being quite annoyed at me sometimes. Not true. That's absolutely not true. No, but I did bother them quite a lot, to be fair. Anyway, I started my my path in 2016 as an undergraduate student in specializing in biotechnology. I knew I wanted to do biotechnology. There weren't many universities offering it. I didn't have really good grades coming out of high school, or A levels rather. So, you know, the University of Wolverhampton was a great fit. So I started doing that. And by the end of the first year, I was actually also a student representative. So on top of seeing a lot of my lectures during lectures, I also went and complained about stuff for students on behalf of students. No, that's not true. There wasn't a lot to complain about, to be honest. You know, it's having also the opportunity to have that aspect of the university life in which you can share your experience and actually your issues that you have maybe in the classroom with other students, also with the professors, is very, very positive, I feel like. And every time that we did discuss something, stuff either did get resolved, or there was an explanation as to why everything could not be resolved. So at the end of first year, and this brings me, I'm not going to talk about my placement yet, because I did something else before. So between my second and first year, I had the opportunity to do an Erasmus project. So as part of the university, because the UK was in the European Union, they had funds to get students to go to other countries in Europe and get students back. So another great thing about the University of Wolverhampton is that it gave me the opportunity to go for 3 months on placement in Spain. And I did quite a lot of analytical chemistry. I was actually, funny enough, I'm Italian, so we do a lot of oils and whatever. And I was doing analysis for oils for companies. That was quite fun. So I did that for 3 months in between my first and second year. Then I picked up my second year, and I did my second year as normal. And then Dr. Gibson, by the middle part of the second year, introduced the whole aspect of the placement year. I was aware of it, but I was not aware of, you know, everything that could be done or could not be done. So I decided to go for it, really, but I didn't want to stay in the UK because I wanted to experience a little bit more of Europe in general. And I found a position within Inocor Pharmaceutical that Dr. Gibson showed in her presentation, and it was an absolutely incredible experience, not only because of the support that I received from both Dr. Gibson herself and all the members of the careers team that helped me set up the actual placement and make sure that everything was all right with housing and everything else, but also because of the experience itself. And I was basically working for this pharmaceutical company that did drug delivery systems. So we produced synthetic biodegradable polymers. And when these were basically formulated with an active pharmaceutical ingredients, they were then injected into the skin. And after injection, when the temperature of the actual formulation reached 37 degrees, they would they would basically clump together and form a hydrogel. And then whatever you formulated inside of it would be released slowly. We did both that and microspheres, which are basically core-shelled polymeric composites that you can either inject or ingest, and they also release pharmaceutical ingredients with time. I did that for a year. I was completely doing novel research and development. The company was very small. It was 50 employees. So I had incredible contact time with all the different departments, from the actual synthesis to the formulation, to the quality control, to the shipping, administration, and all that part of it, which is very important, as they said, as Dr. Connor said previously, on the actual seeing the importance of that in a workplace, understanding how a company works, understanding why you have to do certain formulations as they are for clients that require specific requirements. And that was an absolutely incredible experience. I did actually 11 or 12 months with that company. And then I started my third year. And because of my love for polymers and everything else, I decided to do my bachelor's project with Professor Radecka. And I think she'll have an introductory video after my talk. Basically with her, I was working on one of the three main areas of expertise of her lab, which was bioplastics. So we were basically feeding microorganisms with waste frying oils or waste oils, and they were converting it in bioplastics. And then we would extract these bioplastics with solvents and then characterize them and see the application that we could employ these bioplastics for. But let me speak a little bit, a couple of minutes as well, in terms of the actual teaching and both theoretical and practical. One of my favorite modules ever in the world during my university career was actually the bioinformatics module that I did with Dr. Mark Morris. It was absolutely astonishing. We did very much things that might have not been completely 100% relevant with my project or with my career path, but that doesn't matter, because it's all about having that understanding of how cells work, how genetic works, and all the components and everything else that did work. I also recall incredible cloning practicals, also with Dr. Connor and Dr. Mark Morris. We had incredible industrial visits with Professor Radecka to Marston's brewery, made by one of the ex-alumni, and we actually saw all the process of beer making. Honestly, who doesn't love beer? So let's go see how they make it. Yeah, it was, you have very much variety in terms of what you have in your bachelor's career. I also did some modules at both an Italian university before going to Wolverhampton and a university in Canada, funny enough. I don't know why I did that, but yeah, life. So, and the actual blend of everything that I found in Wolverhampton was perfect. And I did quite well out of it. Actually my dissertation, this is for anyone that will tell you that the University of Wolverhampton is not high in the ranks. So, your exam grades are not, worthwhile or whatever. I did get my dissertation marked by Professor Alison Smith of the University of Cambridge, and she gave me quite a good grade. So, because of the way that the university system is made or is thought about in the UK, other universities regulate other universities. So the grade that you get at Wolverhampton would be the same that you would get at Aston University, Birmingham, or it's just the teaching that is different. But that doesn't really matter, does it? Because I had incredible experience, and so much so that I actually decided to stay. Before that, I was actually part of the committee that granted the University of Wolverhampton with the accreditation 2 years ago. It was Dr. Connor, I think. It was incredible because we saw the feedback that external assessors from the Royal Society of Biology were giving us, and they were asking a lot of very detailed questions. And I was part of the panel that got to answer those questions, and it was an incredible experience. So as I was saying, I did quite well out of the University of Wolverhampton, and I had the opportunity to actually go and study in London or wherever I wanted. But because of how great it was, I decided to actually stay. I went straight on to do a PhD with Professor Radecka, which I'm still doing. I'm almost at the end of the first year. Can you believe that? I'm also working as part of my PhD for Science in Industry Research Center, which is an incredible initiative that is basically, we do consulting, if you'd like to call it as such, for external small medium enterprises that come to us with an idea, and we help them to develop the idea as well as to actually implement the idea practically. So I get to do that. They support me quite a lot in terms of the fees that are for my PhD, as well as they pay me for it, which is incredible. And obviously, you know, I know each and one of the lecturers at the University of Wolverhampton. They know me. Why? Because as Louise and Laura were saying, if you actually want to make the effort and want to engage, they are super available to help you with any sort of query, any question. You can email them. You know, sometimes you see replies at 2 in the morning. No, that's not true, hopefully. But it's wonderful to have that possibility to actually say, I do not understand something. I'm going to seek for help to the actual lecturer. And, you know, you do your research and you try yourself. But if you need that help, the help is there to do so. So I really urge you to consider this university, because it's fantastic. The actual program is fantastic. The universities are is very well equipped in terms of equipment and staff and everything else, and the lecturers are incredible. So honestly, why wouldn't you? And I think that's it for me. If you have any questions, please do ask.
Thank you, Mattia. That's super. So yeah, I think that Mattia shows that if you get involved, you can, you know, you get, and as Laura said, you get out of it when what you put in as well. But we are all available and happy to give that level of support. Does anybody have any questions that they want to ask?
I will also be stopping by later, so if you think of any questions, just please do fire away or put it in the chat, and I'll try and answer it in the chat.
That's super. That's great. Thank you very much. So what I'm going to do now is just show you a little bit more of that video. And this is actually Dr. Kashir. We're going to talk about Professor Radecka's research, and then we'll actually introduce Professor Radecka herself after this. So let me just share this screen. Can we see that? So I can't see you.
Yeah, that looks good, Matt.
Yeah,
okay.
We can see.
Radecka is available on the 5th floor. Here we are on the very 5th floor of the Rosalind Franklin building. This is the top research floor of the building itself. And here we've got some fantastic facilities. On one side, we've got greenhouses that look out towards the sky, and they're lit all the time. So if you look across Wolverhampton at night time, you can see a sort of pinky purple glow from the top of this building. And this is our greenhouses being lit 24 hours a day to grow the experimental crops that they're looking at here at the University of Wolverhampton. But behind me is the home of one of our fantastic research groups, and that's the Biopolymers Research Group. And the lead of that is Professor Isabella Radecka. So let's go and meet her now.
Hello, I'm Professor Izela Decka, and this is my colleague.
Dr. Brian Johnston.
And we are in our fermentation lab in a Rosalind Franklin building at the University of Wolverhampton. And this lab is a fermentation lab. As you can see behind us, we have a lot of fermentation vessels, and this allows us to carry on our research in an area of biodegradable polymers. So we use microorganisms, and we grow them most of the time using waste material, and we produce biodegradable polymers. But we are in fermentation lab, so fermentation biotechnology lab. As you can see, we have fermentation vessels here. Those vessels are used to grow microorganisms. They're like little houses for them, and we can really provide them with very good environment, temperature, pH, lots of food, and we can grow them in a very good quantity, and we can produce very interesting, valuable products.
Okay, so this laboratory's got lots of different uses. We can analyse different types of plastics here because we work on degrading plastics and making biodegradable plastics to replace those degraded plastics. So in the fermenter, we can grow bacteria that help to break down plastic, and then we can make biodegradable plastic. And this machine here helps us to control the conditions of the reactor, so we can control the temperature with this little belt here. So this can heat up and warm up the vessel. We can also change the pH, and we can also increase the speed of the impellers, which spin around and make the bubbles, so that the oxygen levels can go up and makes the bacteria grow more. So before we start the fermenter, we have to autoclave it in this machine. So we fill up the vessel with water, place it in the autoclave, and it gets heated up to 121 degrees for up to about half an hour, and that kills any bacteria to make sure it's completely sterile. And then we can load whatever we need to load into it, and it can grow without getting contaminated.
Okay, so what we have here is an incubator shaker. So if we want to inoculate and put bacteria into our fermentation vessels, which you've seen before, first we have to make them happy and alive and make enough what we call the starter culture. So we prepare little flasks with a particular growth medium. We put microbes in, and we just keep it here overnight until the microbes will grow. We check if they're healthy, if they are not contaminated, and then this goes to a fermenter for the fermentation process, and as Brian said, for our bioplastics most of the time in this lab, because we produce biomaterials, bioplastics. Okay, as we mentioned before, in this laboratory we work with biomaterials. So we grow the microbes first in a shaker, as you've seen before, then in a fermenter, and then after that we isolate the polymer. And when we isolate the polymer, you can see that some of them, they really like little plastic materials. They behave very similar to plastic materials, but it's fully biodegradable material made by bacteria. And for some of our research, we just use a dirty cooking oil, very dirty cooking oil, which is much different to a fresh oil. And when we use this dirty cooking oil as a food for microbes, they can just produce the bioplastic, the biomaterial. Now, we need to know how good the plastic is. So one of the methods of trying to find out if this plastic is of good quality, we use this machine here, which is called gel permeation chromatography. The biopolymers are made from chains. So polymer is a big chain, and now depending how long is the chain, that reflects on the quality of our plastic. So we have to measure the length of the chain. So we use this machine, which is called gel permeation chromatography, to assess this, what we call a molecular weight, the length of the chains in the biomaterials. So Brian, you can say now.
Yeah. Okay. So to prepare those samples, we take a small amount and we melt it down in chlorophyll, and then it gets loaded into the machine through the cassette deck here. So the sample goes in there, then a tiny syringe goes into the sample, sucks up a sample, and then it passes through this column here. And depending on how long it takes going through the column, we can work out its molecular mass. So on the computer will do a graph and it calculates the molecular mass of those samples. So this is our 3D printer. So we can use our biodegradable polymers, mix it with PLA, polylactic acid, and we can make 3D printer ribbons like this and create shapes like this, or even shapes like human ears. And we've actually been able to, we had a paper a few years ago where we grew human cells on these graphs as well. So it's able to be used for lots of different applications. And the fermenter can also be used to make conditions for human tissue growth on these platforms as well.
Wow, wasn't that absolutely amazing? The work they are doing in this room is literally saving the planet. It's not only replacing plastics that we can't degrade with plastics we can degrade, made from waste that we throw away every day. It's also helping to build the scaffolds that in the future we'll be able to actually grow human organs on. And I think that is absolutely fantastic. I hope you've enjoyed having a little tour around the Rosalind Franklin building today and meeting one of our fantastic researchers. So hopefully we'll see you again soon. Bye-bye.
Okay. That was really a nice video. And I'd like to introduce now the real Professor Inetska. I don't know if you'd like to say hi and add to that. It was quite, it was really interesting. So hi, Isa.
Yes, hello Matthew, hello everyone. I've never seen this video before, so this is the first time I've seen it. And yes, thank you for inviting me for this day today. Shall I just put my presentation on and then tell a little bit more about the
That'd be great, thanks.
teaching and can you see?
Yes, we can, thank you.
Okay, right. So as you can see on that video, I'm a biotechnologist. I'm a professor of biotechnology, but I didn't start with microbiology in terms of my career. So I think Mark said it before, and then Matthew, and then Mattia. I started to study biology, biological science, because I wanted to understand how the cell works. And it doesn't matter really if it's microbial cell or human cell or plant cell. If you understand how the cell works, you can actually use this knowledge and do something cool, make products, make new drugs, fight with different diseases. So my first degree was, I did my degree in Poland, and it was master degree. And the first 3 years, we just had chemistry, cell biology, physics, mathematics, nothing else. And then after that, we had to decide what we want to do next. So I've decided to do my degree in histology and anatomy, and I was working with brain tissue. So I was investigating the effect of heavy metal pollution on function of brain cell, tissue or organ as a brain. And then after that, when I finished, I actually started my job at the Polish Academy of Science in a Polymer Institute, and that's when I developed my interest into biodegradable polymers. And then I started to work at the university as a teacher in biochemistry, and I did my PhD at biochemistry, but my PhD was focused on microbial biotechnology, and particular on microbial growth and production of those biodegradable polymers. So then I worked a little bit in Germany, a little bit in Denmark, and then I ended up in Wolverhampton, and I joined the team of microbiologists. And I sort of transfer my research into teaching and research. And that's why I sort of started developing more and more the work with biodegradable polymers. So instead of in terms of teaching at the university, I teach on undergraduate and postgraduate levels, and I teach everything what is related to microbial applied biotechnology. So, for example, I was involved in first year teaching, which is Introduction to Microbiology. Then I was teaching, I'm teaching applied to microbiology on the second year, and on the third year, I'm teaching module which is called Microbial Biotechnology. And those modules will teach you about sort of traditional biotechnology and modern biotechnology, and we would have lectures in modules. We would have guest speakers, as mentioned before. I try to always organize some interesting industrial visits for students so they can see not just the lab at the university, which is relatively small, even we think our labs are quite big, but when we go to industrial scale, they actually feel quite small. So we go and see the real industrial place where a lot of actually our graduates work. So they take us and show us around. And I also invite guest lecturers, guest speakers. And this year, for example, we had, again, a very good team of people. And some of them were talking about very hot topic, the production of COVID vaccines. So we try to cover different areas. But in terms of my research, as you could see on that video, I'm trying to produce different novel biomaterials in a cost-effective way and in a sustainable way, and then transform them to what we call the highly valuable products, which could be food products, could be pharmaceutical products, or could be just a plastic packaging material product or drug delivery products. And I'm also interested in wastewater treatment, and a lot of teaching I do is about wastewater treatment. And microbiology always fascinated me, and microbes are a really good sort of group of organisms to work with. They're very adaptable, they're very smart, they can survive in a very difficult environment. And sometimes this is good for us because they develop different pathways, metabolic pathways, which we can utilize. And we can then use the microbial activity or products in either environmental application, pharmaceutical application, food application. So how do we put microbes to work? Well, as I said to you before, from the teaching I do in microbial biotechnology, we talk a lot about production of alcoholic products, cheese, bread, or other food production, but we also talk a lot about production of antibiotics, vitamins, different enzymes, which can be used either in food industry or even for Mark's cancer research. Can use those enzymes, thermophilic enzymes for PCR, for example, but also some food additives, which we can also produce using microbes as this key sort of factor. We cultivate the microbes most of the time in a fermentation vessel, so we can do it in small shake flask, but to make it more sort of industrial and applicable, we transfer the production of microbial culture into fermenters. And on this movie, you were able to see how we do it. So we've got different size fermentation vessels when we can grow our bacteria, and we can control different environmental parameters like pH, temperature, agitation, aeration. And that has influence on the physiology of the cell. And because of this very sort of strict, under strict environment cultivation, we can obtain a particular useful product. And here you can see, this is the lab where this little film was done, but you can see this is the range of the fermenters here. And my students on the level 6 or third year during the module microbial biotechnology, we have a session in this lab, and also master students and PhD students work in that laboratory. In terms of my interest, as you could see from the movie, my team is interested in biopolymers, biopolymers which can be used for particular novel biomaterials. But of course, we have to think about the performance of the material, and we have to think about the price of the material. So we have to very carefully choose the biodegradable biopolymers. The ones which are of our interest is bacteriocellulose. It's a protein-like polymer, polyglutamic acid, and the family of polyesters, PHAs. And those are the ones which you can very soon see on the market. And some of them are actually already on the market. So the first one is the polymer of polygamma glutamic acid, which we work with. And actually Matthias' project relates to a production of polygamma glutamic acid. This particular polymer is produced by a bacteria called a species of Bacillus, very sort of common group of bacteria. It's a polymer which is just made of glutamic acid, amino acid, and you can see it can be used as a food material. Natto, it's nothing else like fermented soybean food product. Not very nice, I've tried it, but it's okay. But the sticky material here is the polygamma glutamic acid. So what you can see here, and this is the formula. And why this polymer is important? Because it can find a lot of different applications. So we can have medical application, which is a drug delivery system, biodegradable scaffolds, and you can see that you can make biodegradable scaffolds, for example, from this material. It can cryoprotect our food. It can have influence on taste, on the texture of the material, but also it can be used for other applications like wastewater treatment. The big sort of length of the chains can very easily detect metals in the wastewaters and can sort of remove the heavy metals very quickly, and then the water can be treated further. Also, now it's very popular in cosmetics because it's a hydrogel, so it's got very good hydrating properties. So you can find actually in Boots, they sell 4% polygamma glutamic acid now as a cosmetic, as a remedy for beauty. We've used this material as a protective agent for probiotic bacteria. A lot of people use probiotics to be more healthy. They help with digestion and have other different functions in our body, but a lot of probiotic bacteria would die when they pass through either production line, so during freeze drying, then you have to store it in a shop, in a bottle, in a low temperature, and then eventually it goes to our stomach where the pH is below 2, and of course a lot of cells will die. But what we found out that if we cover those cells with our hydrogel of polygamma glutamic acid, they can stay viable, and you can deliver a lot of the cells into your intestines, and that could have a positive effect on your health. And we were very happy to find out that it was a very good innovation, and we were able to file a UK and European patent. So now we are working with companies towards exploitation of that patent. Another polymer which we are interested in is bacteriocellulose. You think about cellulose, you think about plants, but bacteria can also produce bacteriocellulose, and you can see you can grow it in a sort of static culture or in a fermenter. And you can see this sort of jelly layer here on the top. This is our bacteriocellulose. When we clean, it becomes transparent, beautiful hydrogel. And when you do electron scanning microscopy, you can see that the hydrogel is nothing else like little, quite long chains made of glucose. And then the water is between those fibrils of the polymeric material. It's a very good material in terms of the fact that it can be moldable to, for example, if you use it as a wound dressing material, it molds to the skin. It's chemically pure. The gaps in between allows us to load it with antimicrobial agents. So we worked with together with Dr. Gibson, who was telling you before that one of our PhD students is working with bacteriocellulose and trying to make a novel wound dressing material. And again, application, so medical application. So we are in that category, what we're trying to achieve. So we're trying to achieve different type of wound dressing for especially people with severe burns, for example, because it's a hydrogel. It's got not only it's moldable, but it's got this nice cooling effect. So it relieves a lot of pain from the patient. But what is good as well, that you can make this polymer, you can train the microbes, the bacteria, to make a particular shape. So here you've got an example of the tube, which can be used for cardiosurgery. This particular material also can be used as a food material. Some people grow this bacteriocellulose and then submerge it in a juice. And coco de nata, it's a delicatessen. It's like ice cream, cold, so you can make this as well. And some people also are exploring the packaging or cosmetic or textiles application for this particular polymer. When we do microbial biotechnology practical on level 6, students produce this particular polymer and design a wound dressing material, and then we test antimicrobial activity against pathogen. And even during the COVID time, would you believe, we were able to have some practical sessions, small groups, obviously with distancing, but we were able to do it, and hopefully this year we will be able to run it successfully again. And then the last one is the microbial bioplastic. So as Matthew said, we are working towards development of biodegradable plastic materials, this particular polymer can be made by bacteria from waste under stressed conditions. So what we're trying to do, we're trying to be quite unpleasant to microbial cells. So we either don't give them enough oxygen or not give them enough nitrogen, or the pH a little bit is lower than expected. So we put the microbes under stress conditions. And then when we put them under stress conditions, they suddenly start to produce granules inside the cell, so accumulate those granules of the polyhydroxy acids, and then we can get ththose granules out, and we can produce different materials from it. So again, we can use it for medical application. Oh, I'm sorry, I just went too far. Where is the one? I can't see the. It's difficult to go back. So you can see the medical application. So we can produce biodegradable scaffolds and grow cells on it. Again, we can use it for drug delivery systems or different type of implants. But also, so this is the very pure form, but also we can make agriculture mold film. And we've done it in our lab. We published a very good couple of papers from it, and also we can make a biodegradable packaging for food. And as you know, the problem with packaging and plastic is terrible nowadays, so we're trying to make sure that we can produce plastic trays like this, which in a compost environment can be fully biodegradable to CO2, water, and microbial biomass. And this is the mold film which we produced. So we've added herbicides with very low percentage, and we were testing if this particular mulch film can release the herbicides and can prevent formation of or growth of weeds, and we succeeded. And we also had a lot of data about the plant growth, and we found out that it doesn't affect the plant health. So it's a good application. And then you can see those are the plastic trays which we produced. They also can have nice colors, and those colors are also natural. So it's nothing which will affect the biodegradability of this particular material. And this is particular of our interest now, because as we know, it's a lot of problem with plastics. Lots of microplastic is produced, and this microplastic will stay with us forever. And it was found out that about up to 90% of waste or plastic waste in the seawater or oceans is a plastic, and you can hear the news, I suppose, every day. So this is a picture of the polyethylene bag which was found at the bottom of the Marina Trench, so the deepest sort of place on our planet. So that's why we're trying to make sure that we can, in our lab, with my team of undergrad students doing their project, master students doing their research project, and PhD, we try to achieve, we try to produce new materials, biodegradable materials, and we use waste product, agriculture, industrial waste, or even plastic waste to produce this material. And we publish the data, we present the data through the conferences. Actually, our last paper was released 2 days ago, so we are very happy.
That's great. Congratulations,
Thank
Lisa.
you.
Thanks, everyone. Would you stop sharing your screen for a second, Isa, just so I can put one last slide up? I realised that was really interesting, but I do realise that we are close to time. So I know that Mohammed had a quick question. I don't know if you've still got it, if you wanted to ask that, or anybody else has any questions they want to ask. Please feel free to ask them now. Okay.
Hi. Just a quick thing. I mean, the questions that I did have in mind, I mean, collectively, everybody's pretty much answered them. So I just wanted to say thank you very much for that. You know, it's been a real eye-opener as far as what the University of Wolverhampton has to offer to all new progressive students. So yeah, just wanted to say thank you for that. You know,
that's great. No, thank you for attending. That's really good. If you have got any other questions, you can always find our emails online. I'm happy to answer any questions by email, and you've got the Chatify session next to ask professional services, and they can always direct questions back to us. Could I also ask you, can you see my screen on this survey that I put up? Can you see that? So there's a survey there if you'd like to complete that, just to let us know what you thought of today. That'd be great. But as I said, otherwise, I'd like to thank you for attending, and just get in touch with any questions you might have. Hopefully we've shown you what the courses are about and helped you to make the right decision. And thanks everybody else for attending. That's really good. But I think you'll be put back to the main meeting now. But thanks everyone.
No problem.
Thank you.
Thank you. Thank you, everybody.
Okay, thanks. Bye-bye.
Bye.
Thanks.
Thank
Bye.
you, everybody. Thank you.
Thanks. Bye.